Okay guys, let's start the lecture on cestodes. So, quick review, we've covered roundworms, right? Those included our roundworms themselves, hookworms, whipworms, and then our GI roundworms, our lungworms that were round, our bladderworms, and eye worms, etc., etc. Heartworms are included in roundworms. Then we covered heartworms, but of course they're in that group. And now we are starting to move on, finally, into cestodes, okay? We're going to cover them specifically with small animals, okay? I've grouped the large animal parasites together by their large animal species group. All right, so this will be just the small animals. So let me click here to get this presentation up and going. All right, there you go. There are the names of the different types of cestodes, i.e. tapeworms, that you need to have study material made for. Okay, remember I do need to see your study material after spring break in order for you to receive points back on the test. Not a lot of people received points back on this last test because their study material was lacking. So keep in mind, this list is an excellent one to go off of that you need to make a note card for each one of those bullets at minimum. Really, you need to separate some of those bullets, especially the last one, the Spiometra and the this lovely one, need to be separate note cards. You need to have a photo, most importantly, at least one photo. Should be more, honestly. The scientific name, the common name, and important facts. Oops, sorry. There we go. So let's start out with the common tapeworm of dogs. This is Dipylidium caninum. It's also known as the cucumber seed tapeworm, the flea tapeworm. There's lots of different names for it, but honestly, what you're going to hear it called out in practice is just the tapeworm. Okay, it can affect both dogs and cats. We've covered this one a little bit before, so if we were in class live together, I would say, what is its intermediate host? I hope you all just said flea. Yes, the intermediate host is a flea. So, what does that mean to you when educating clients about this parasite? I hope someone just raised their hand. I'm calling on them. It means they can only get infected if they eat a flea. Correct. Very good. So if the animal eats another dog's stool sample, okay, their poop, say you're at the dog park and your dog goes over there and eats somebody else's poop, can they get Dipylidium caninum? No. That's correct. Excellent job, everyone. No, you cannot get Dipylidium caninum by eating another dog's stool, even if they are infected with this parasite. You must eat a flea. Okay, so this is an indirectly transmitted parasite. Inside each one of the segments, so right down here you see a picture of the worm. Okay, see I've got my mouse cursor running over it. This is what the adult parasite looks like. And its body grows and forms these segments that are known as proglottids. Okay, there's the word right there, proglottid. Okay, these proglottids break off and are passed in the feces. Okay, they move like a little inchworm. Quite disgusting. Inside each one of these proglottids are hundreds and hundreds and hundreds of eggs. Okay, and the eggs we refer to as hexacanth embryos. See, just like a person's egg is an embryo, same word there coming back. So the animal defecates with these proglottid segments. The flea lands in the poop. These little segments will kind of break down and bust open. The flea ingests that hexacanth embryo that then matures in the flea. Remember, this is an intermediate host, so the parasite must undergo a change while inside the intermediate host. And then the animal, the dog or cat, ingests the flea. So you may be thinking, well, why in the world would my cat eat a flea? Your cat doesn't realize they're eating a flea. Your cat is using its mouth to scratch itself because the fleas are making them itch. So the animal whips around and gnaws at itself with its front few teeth to try and relieve that itch and ingest the flea. there is a very, very zoomed in microscopic examination of what the head of this tapeworm looks like. You see all these segments are very, very narrow the further up the worm they are, but as the worm grows, these will separate out into those little proglotid segments that will eventually break off. Okay, so this right here is what we refer to as an egg basket. Okay, sometimes people call them egg packets. It's just the term for all those embryos are together inside of this packet. Okay, so if we have a bunch of embryos all stuffed together, do you think that that's going to be heavier or lighter than one ova by itself? I hope you were thinking along the lines of heavier, because that would be correct. Okay, there's lots of embryos in there, so it weighs it down more. That being said, what does this mean in practice? It means that it is less likely that we are going to see these egg packets on a fecal flotation because they're really heavy. So it's possible that they may sink even despite our fecal solution with a higher specific gravity. Okay, so often this parasite is diagnosed and treatment provided to the animal by visualization of these proglotid segments. So here we go. If we were to look at each one of these hexacanth embryos, you would see these little hooks. Okay, here's an example of what you might see on a microscope. Again, you'd have to be very zoomed in. We're talking about at least using our we're beyond our 4x at this point, beyond our 10x, probably at our 40x, and that's where you would maybe see these lines, the striations, the hooks, so to say. Okay? There are 6 of them, however, typically we only visualize 3. So what are we going to see in an animal that has Diploidium caninum? Okay, typically the only thing the owner is going to notice is the little bits of rice, or they look like cucumber seeds, inside the feces. Okay, so the animal's going to defecate and they're going to see the little white bits. Those proglottids are pretty sticky too if you touch them. You don't realize they're sticky, but they stick to the hair around the rectum of the animal. So a lot of times in clinic when I've actually found this, the animal was not defecating, but I was doing my little pre-exam before the doctor came in, and when looking at the rectum, I noticed one on the hair near the cat's rectum. Okay, or for the dog, you'll often notice it a little further down, maybe by their feet. A lot of dogs love to kick backwards with their hind legs after they urinate or defecate. It's a way of scent marking the area. And so sometimes you'll see those little proglotid segments on their feet. So make sure you just check around to see if there's any of those little segments. That would be how you would find it in clinic, but typically the owners will call in and they'll be like, I don't know what's going on, but I swear it's like my dog's pooping rice, but he hasn't eaten any and I don't know what to do. So there you go. That's the diagnosis. This is one that humans can get. Now, we would also have to ingest the flea, which I know sounds crazy, but how many of us sleep with our pets? Yeah, I know I do. My cats get to sleep with us. The dogs typically don't unless it's their birthday. When it's their birthday, we let them sleep with us. They get to have us all to themselves. The other dogs get put up and they get in bed with mom and dad, and we tell them their adoption story and how we got them and that we love them, and they get to spend the night with us by themselves. Very sweet, isn't it? But we sleep with our pets. How many of you sleep with your mouth open? You might not know it, but if you are a snorer or if you are somebody that gets into some really deep REM cycles, I know this, I've caught myself like this a few times, your mouth, the muscles in your jaw will relax and your mouth will actually drop open. So it is possible to ingest a flea unknowingly. That being said, it is of course rare, so don't create panic and fear in the owner, but this is something that they could potentially be infected with, so let's get it treated, right? It's easy to treat for. All right, we talked about how we don't see the eggs on fecal float because they are heavy. They're laid in those packets or baskets. If you do have a proglotid segment that you guys find in clinic, take some slides and squish it in between 2 slides so that you can see the eggs. Now, also, what test do you think we could perform if this is a heavy egg that we might get to see. So let's say we might get to see the embryos. So let's say the owner said they saw some white worms and they pack up the stool and they bring it in and we don't see any of those worms maybe. But we're going to do a fecal float anyways and it's negative. So now the doctor's like, all right, what do we do? Well, first off, the doctor would probably know what to do. But second off, you could do a direct smear. Okay, again, no promise that it will get it, but if the egg packets are visible in the poop, just they're sinking to the bottom, then we could do a direct smear and potentially see them. We could also do what kind of test? There should be at least one group in the audience that knows which test I'm talking about. Sedimentation, correct. So that would be where we would centrifuge our sample down and then be looking at what is forced to the bottom of our centrifugation tube. Okay, so we would be intentionally examining that top layer of sediment at the bottom of the tube after we've centrifuged it. Okay, and again, the only way we're going to see these eggs is if that proglotid dried and opened up, okay, and then those hexacanth embryo baskets or packets were released. So, as you can imagine, this is a hard one to diagnose under a microscope. All right, but we can treat for it. There's an easy medication, praziquantel, that we would use to treat for it. It's just a couple-day course of medication. But the most important thing you must tell the owner about this parasite is that they need to take care of the fleas. Okay? And this is when owners love to argue because they're like, my dog does not have fleas. Well, at some point your dog ingested a flea. So think back, maybe they did have fleas and you don't want to admit it and you put them on preventative and now we're seeing the signs of it. That could be possible, all right? I have seen tapeworm segments turn up weeks after an animal was on a preventative and not having any more fleas. But keep in mind that if fleas are somewhere present in the environment or on another pet, everyone is at risk of reinfecting themselves. So flea control is an absolute must with this parasite. So there you go. This is a cat's tail, and you see that tiny little white segment. When they dry up like this, they turn from that white to a darker color. Look how tiny that is in comparison to the penny. So let's see this video. Hopefully this will work, opening it up on my Camtasia software that I'm using to record this video. So give it just a second to load there. This should be a video of the tapeworm segment itself moving. There you go. It will get long and thin and then squish back up. So you see it? There it goes, squished up. And here we go. It's going to stretch out. Crazy. Absolutely crazy. Kind of moves like an inchworm almost, but instead of the body inching up, it collapses. See, there you go, look at that. Oh, that's crazy. It's like flubber. Oh, so gross. Oh no, don't touch it. Ew, ew. They want to show you size comparison. Ew. Oh, we lost our elevator music. Oh, don't worry, it started over. There we go. Oh, that's so gross. And they only move like that once they separate from the main worm. Okay, so that's not going on in your pet's rectum, but oh, crazy, isn't it? Crazy, crazy. All right, where's our PowerPoint? Right here. All right, so let's move along. Now we are going to the feline-specific tapeworm. Okay, so remember, tapeworms can get Dipylidium caninum. All right, and yes, we would still write it down as Dipylidium caninum. They can be a definitive host of that. However, they do also have their own form. So this is Taenia taeniiformis. Isn't that nice that you only have to memorize 6 of those letters once because they're in both words? So Taenia taeniiformis. This is the feline tapeworm. So the definitive host is the cat. There is an intermediate host, as with most of our cestodes. This time it's a rodent. Okay, so what do cats love to do that go indoors and outdoors? They hunt, just like that dear cat video where the kitty cat laid, he says, I have brought her many, many treats, and judging by how quickly she picks them up, I know she appreciated them. So yes, they will bring us little rodents that they have eaten, and that is how they ingest this parasite. So the place it loves to live is the small intestine. You guys remember our review of the intestines? Starts at your mouth, goes down the esophagus into the stomach, into the small intestine, which is made up of the duodenum, the jejunum, and the ileum. Flies past the cecum into the large intestine, which is made up of the ascending colon, the transverse colon, and the descending colon. All right. When the animal defecates, the cat defecates those proglotid segments, then the rodent comes along and eats those, and then it undergoes transformation while in the intermediate host, and then the cat ingests the rodent. So what are we going to see? It's the exact same as our Diploidium caninum. You're not going to really see any symptoms except for maybe some proglotid segments in the feces. However, it's hard to diagnose because this cat is going to be our indoor-outdoor cat, right? So unless it also has a litter box inside, you may not notice it. Let's see here. Again, same treatment. Ditto, ditto. Isn't that great? One of our best ways to prevent this one is to get the cat to stop hunting. Although that is really difficult for an indoor-outdoor cat to convert them to an indoor only. They have a lot of those learned behaviors that are very hard to give up. So it is a tricky thing. It's almost kind of cruel to take an outdoor cat and then try and make them an indoor cat. Just because their world completely changes. I mean, it would be if you were taken off planet Earth and put on a different planet, you know, because everything would be different. Your normal activities would cease. Your environment completely looks different and it feels different and smells different. So it's a tricky thing to take an outdoor cat and make them indoors. There are the eggs. So what do you notice with this egg packet as compared to the last one we noticed? You may be saying it's a little darker. That's true. The hexacanth embryos are darker in color. What do you notice about the shell? Do you see a shell? There is not typically seen a shell. So this one reminds me more of like a cluster of grapes. Whereas if we go forward, bear with me here, to this one, you see the shell? Typically there will be an envelope that holds in the hexacanth embryos of the Diploidium caninum. Whereas the Taenia taeniiformis, typically the embryos cover up the edge of that sac that surrounds them. Okay? Do you guys see some of the hooks in here? We've got some right here and here and here. They're hard to see, but they are there. What about the ova itself? So we're talking about the hexacanth embryos here on the right side of our screen. What do you think about the shell? Is it thick or thin? It's thicker, right? We can see where it starts and where it stops. What about the shape? Yes, it's a circle. Good job, good job. What about the center material? Does it take up all of the shell? Certainly does. What about the color? Does it allow a lot of light through? Or the transparency, excuse me. It is less transparent than some of the ones we've seen, right? There's a lot of color in there. Not a lot of light comes through since that center material takes up the whole shell. Keep in mind these are very, very tiny if seen on the microscope by themselves. All right, so let's talk about some other species. All right, this should take you to a pronunciation of that. So other Taenia species. Okay, I do want you guys to be prepared with these names. Okay, there are more questions that I would ask in your multiple choice than identification, okay, because their eggs are going to look very similar, the ova. All right, but you need to know the names and as far as what the intermediate host would be. Remember I told you I love matching with the intermediate hosts. So these other tapeworm species are not going to be zoonotic. And all of these on this page prefer the dog, okay, or wild dogs. So we have Taenia pisciformis. That one is the most common on this list. The intermediate hosts there are rabbits. So this is our dog who loves to chase and bring back rabbits from fields. A lot of our newer neighborhoods here in Texas are invading rabbit areas, so they're completely inundated with rabbits in the neighborhood, and dogs are chasing them, catching them. We're seeing a little bit of this in the clinic. Taenia hydatgena. This one is much more rare in dogs. It's going to be where dogs would consume a ruminant. So this would fall in with our cattle, goats, any of those ruminating species, okay? You may think, when in the world would a dog eat a cow? Well, if you're in the country and a cow dies, you can't just put it in your car and drive it to the vet, okay? Again, this is a very large animal. So typically what farmers will do is they'll use their tractor to pull that cow out to the back of their pasture, and then they'll just let nature take over. So coyotes and things, which fall into our canids of that definitive host, will consume the animal. Wild boars will eat a lot of it, and then the bones are left. Well, you can keep in mind that Scruffy, the family farm dog, also makes it back there to have a little bit of a treat or to gnaw on some of those bones. So that's how they might pick it up. Taenia ovis is also very rare. When you hear ovis, you should immediately think sheep. Okay? Think back to your medical terminology that you had last semester in intro. So we call this one, as its common name, the mutton tapeworm of dogs. Mutton is what we would call any sheep that we eat that's over the age of 1. When we eat lamb, that's baby sheep, okay? Just like veal is baby cow that we eat. So mutton is the meat after the animal has matured, so to say. So it's no longer a baby. The meat's usually a little bit tougher. So this is known as the mutton tapeworm of dogs, and the intermediate host is, of course, a sheep. So what do we see with these? It's going to be ditto, 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 ditto. Isn't that wonderful? It's like less studying for you guys. I get excited when that happens. So the diagnosis is we're going to see proglottids. Okay, these tapeworms are the same as far as their body structure. All right, I hope you learned that in intro about their scolex at their mouth and the way their bodies are formed and how they release their proglottid segments, things like that. You should have covered all of that in intro. If you didn't, or I'm sorry, not in intro, in lecture with Dr. Bader. If you didn't, somebody email me and let me know. But hopefully you covered it. So this is going to be ditto everything we learned about Diploidium caninum, okay? The one difference is we see more striations in the thick shell of this hexacamph embryo. But again, guys, it's going to be so difficult for you to identify exactly the details of this egg because it looks just like the others. So I like to ask multiple choice questions. I'm going to be honest with you about that. If I do have an identification, you may look at it and think, that looks like all the others, right? But what am I going to have in that bottom corner of your screen? I am going to tell you what animal's poop it came from, okay? So maybe I'll say you're working in a large animal practice where a sheep farming owner brought in their dog. All right, I hope that tipped you off and you were thinking Taenia ovis. All right, something like that. I like to give you those hints, not because I like to give you hints, but I give you those facts that you're going to have in the clinic to help you solve these problems, okay? When you are in the clinic, yes, you're going to constantly be having to critically think, okay? You're going to have to be thinking, all right, this could be a couple of things, but let me think about the history that I got from the owner. What do I know about these people, okay? So, I like to provide you with those facts that you would have in a clinic. All right, so let's talk about multiceps suralis and multiceps multiceps. Isn't that one lovely? Multiceps multiceps. You only have to memorize the word once. Beautiful, beautiful. For all you spellers out there that are struggling, you should rejoice over that one. Okay, so with both of these, the definitive host is a dog or a wild dog. Have you guys noticed the predilection site for all of these tapeworms we've covered so far is the small intestine? That's what the SI stands for. All of them have had an intermediate host. The scientific name has changed only because of the definitive host and the intermediate host. So I hope you have picked up on what to study so far. Okay, those of you that have this project for your group project too, I hope you're noticing the things that you want to hit on. Okay, so multiceps cerealis, or cerealis, some people say, depending on if you pronounce that middle I, cerealis or cerealis. The intermediate host is going to be a rabbit. Okay, for multiceps, the intermediate host is going to be a sheep. Okay, I know that multiceps cerealis with a dog and the rabbit is the intermediate host may make you think Taenia pisciformis. Yikes, right? It's the same thing. They both have the rabbit as an intermediate host. So maybe I wouldn't ask that question because it would be too confusing, but you need to be aware of both of those. Okay, multiceps is going to have a sheep as the intermediate host. One thing that we see with this parasite, let's see here if it goes over it. It doesn't on the next slide. Okay, we will see cysts in the brain form, okay, as a result. So that's what this image is of. You can see right there, okay, this is towards the back of the brain, near the brain stem, that we are seeing a cyst. Okay, so it's filled with liquid. Typically cysts are filled with liquid that can be drained out. And that is one of the side effects of this parasite. So often we will see it post-mortem, okay? So ditto, 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 everything that you've learned, all right? I hope that's easy and not frustrating for you guys. The one new thing that's on this slide that I want to point out, which is true of any of these parasites we've studied, is that when you have a heavy enough infection of those adult parasites, of course they can cause an internal obstruction, an intestinal obstruction, depending where they're at, okay? The small intestine, the structures in the small intestine are typically more limited in diameter. So if you have a parasite that's in the small intestine, it takes less of it to cause an obstruction. Whereas the large intestine, the diameter of the vessel, not just the length, we call it the large intestine because of the diameter, okay, is typically wider. It has a little more stretch to it. And so it takes more of the parasite to cause an obstruction there. And also the small intestine is closer to the stomach, right? So an obstruction there, the animal's going to stop eating a lot faster. They're going to show some more symptoms of, maybe some vomiting with an intestinal obstruction. But again, that's with multiple adult parasites. So they would have to be exposed to the intermediate host in large quantities, okay? All right, so now let's talk about one that I promise you is going to be on your test. Just saying it right there. Hymenolepsis species. Okay, there's two here, and you do need to know each one. Okay, so you need to be able to spell Hymenolepsis nana and Hymenolepsis diminuta. All right, diminuta. So here we go. The definitive host is rodents. This does include your hamster, your mouse, your rat, okay? It includes those. Technically, guinea pigs are not in the rodent family, so would not include your guinea pig. But this one is zoonotic and covers the most, not covers, but accounts for the most common cestode infection that humans get. So it is more likely that you will get a tapeworm from a rodent than you would from your dog or cat. Why, you might ask? Well, if you live somewhere where there is a rodent infestation, okay, I think back sadly to when my grandmother passed away, we went over to tell her boyfriend who lived by himself and he was elderly, he didn't have any kids, and we went over to his house, which we didn't do very often. He was a very private man. But we went over there and she had been sick for a while and we just saw how his house was in such disarray. So while my mother talked with him, my aunt and I went to cleaning up his house. I never saw a mouse, but the amount of droppings that I saw from those mice was stomach turning. Okay, and this is here in our country in an apartment complex that regularly gets treated. It's just, he's elderly, his vision's failing. I mean, I guess they were all around. And you want to guess where I saw the most droppings? I bet you some of you are already guessing. In the pantry, in the kitchen, in those cabinets. I mean, it was evident. It was around where the plates had been, things like that. Oh, it was just so sad. And that's in a first world country. We're not even talking about the third world countries and things like that. So, and this one also, the reason why it is so infective to people is that in the Hymenolepsis nana species in particular, it can be directly infective. So that means that you can get this tapeworm without consuming an intermediate host. Okay? So you can get this tapeworm from just getting some of the embryos from the feces. All right? So no intermediate host for the Hymenolepsis nana species. The Hymenolepsis diminutive, yeah. Struggling with that one. Just make sure you spell it right. N-U-A-T. Wait, U-T-A. There we go. That one does have an intermediate host, okay? And there's a variety of them. Fleas, flower beetles, and then just various different arthropods. So, but you need to know that there is one tapeworm in this class that is directly infective, okay? And it is zoonotic. So, look at this egg. It's very different, isn't it? It's very different. First off, it's shown by itself. There's no egg baskets or packets. That's because we don't typically see the Hymenolepsis species in packets, okay? The ova are by themselves. Second off, what do you think about this parasite? Does it allow a lot of light through, this ova? Yeah, it allows a lot more light through. What about the shell? Is it thick or thin? We can see where it starts and stops, but compared to the other tapeworm parasites we've seen, this shell is pretty thin, right? Is it smooth or rough? Smooth. Are there striations that you see? No, whereas some of those other tapeworm hexacanth embryos had striations along their shell. Does the center material take up all of it? Sort of, right? This one's a little confusing. I love this picture over here because you can see where it kind of gathers and stretches off. Like if you've ever cracked an egg, a chicken egg, you'll see that there's a spot where it kind of attaches. Well, this one does at both ends. Okay, so to me it looks like almost one of those little Werther's Original caramels, right? That have been twisted at both ends, or like a peppermint that's got the wrapping twisted at both ends and then put inside the shell. You can kind of see it there. All right, it's because this is technically a bipolar egg, but the poles are inside of the shell. Okay, remember we learned that term when we were covering our whipworms. The poles, bi meaning at both ends, polar, poles at both ends, those little knobs that are there at both ends. Also, since this is one of our tapeworm eggs, it does contain those 6 striations, although you may only count them as 3. Like, see, I like this picture because it kind of shows you how they can look merged together if you see them on a microscope. Okay. As far as clinical signs, these are rodents, so we're not caring as much. However, you may see rats in the clinic. Rats are very intelligent. A lot of people absolutely love to have them as pets. Okay, so what we might see with the rat is we may see diarrhea, weight loss. They also can get blocked because of this parasite, so they may not be eating as much, things like that. Okay, so let's talk about the Echinococcus species. Okay, this one is very important, and we are going to cover it again when we get to large animals. But this is the only tapeworm we're going to cover that has 3 segments. So it does not let off proglotted segments, okay, like the other parasites do. This worm is only 3 segments long. Okay, how many segments long is it? 3! All right, is there lots of worms in this PowerPoint that are 3 segments long? No, there's only one worm, and it has 3 segments, okay? The other worms have more than 3 segments. So if I ask you, the shortest worm that we covered in the Cestodes PowerPoint had how many segments? The answer would be 3. All right, so Echinococcus granulosus. This one is one of the, what we would call the hydatid tapeworm, okay? The definitive host is dogs, and again, it does have an intermediate host of sheep, cattle, or any other herbivores. So that includes deer, anything that might be grazing, antelope, things like that. Okay. Echinococcus multilocularis, okay, is the multilocular hydatid tapeworm. The other one is the unilocular hydatid tapeworm. With this Echinococcus multilocularis, the definitive host is a cat, and the intermediate host is a rodent. Okay, so with this one, I want you guys to know the Echinococcus species. Okay, Echinococcus species. And remember what that adult worm looks like. So what we're talking about with the hydatid cysts, okay? Cysts, again, are going to be things that are typically filled with liquid, okay? And they cause issues because of how they push things out of the way and where they're located. All right, so this one, the Echinococcus granulosus, we see that cyst form in the liver or the lungs or other vital organs. Okay, a lot of different cultures eat organs of animals. Again, the definitive host is going to be the dog, so it has to be a culture that would eat dogs. Some do, but it can be zoonotic. Okay, then not so much if you eat the hydatid cyst, excuse me, I don't mean to make it sound like that. You would get it by eating an intermediate host, but we will see some of those hydatid cysts in the intermediate host as well. Okay. The multilocularis, those cysts will form in the liver and the lungs. They're very fast growing and invasive. So they typically will kill the intermediate host quickly rather than the granulosis will let them live a little longer. As far as the definitive host, they also get these cysts. And the only way we can treat that and get rid of the parasite is to actually remove the cyst. There's some pictures. Here in our country, we're not seeing this often. Okay, thankfully we're really not seeing it often. Look at how huge that cyst is compared to the brain. Whew. All right. So we're not going to see those proglottids in the poop because the worm has how many segments? Very good, 3. So we would actually do something called purging the intestines. So you clear out the animal, whether it be by kind of like one of our colon cleanses, so to say. They have similar ones for animals, okay? And then we would give them a liquid and collect it as they defecate it out, and then look for in the mucus or the defecation of that clear liquid for eggs and things. But honestly, this one is not one we're seeing very often. It is one that is monitored and needs to be reported if found here in our country. So that's kind of interesting, but I just want you all to really focus on the Echinococcus species and that the adult worm has 3 segments. Okay? All right, we're getting near the end here. This one is one I'm not even going to try to pronounce. All right, it's a tricky one to pronounce. It actually falls into our category of trematodes, but because of its appearance, it looks like a trematode, but it's actually known as a pseudo tapeworm, okay? So the definitive host here is going to be a dog, a cat. Yes, people can get it, so it is very zoonotic. There's 2 intermediate hosts, which is something you're going to see next week that's very common with our trematodes. The first intermediate host is any of our aquatic crustaceans, okay? So crabs, snails, things like that. Then the second intermediate host is going to be a fish, okay? So you can see how people can get it. All right, get ready. You're not going to want sushi after next week. Predilection site is the same, small intestine. Okay, one thing that we really see with this is that the parasite likes B12. Okay, same thing with a lot of our trematodes that we're going to cover. And B12 is what I heard one doctor refer to as the groceries for your blood. Okay, so red blood cells need groceries essentially to eat and be strong and make more red blood cells. And B12 is one of those. So if your body is drained of B12, you will actually start to become anemic. Not because you're losing red blood cells, but because your body doesn't have what it needs to make more. So we classify that as pernicious anemia, okay? And so that is when you are deficient of B12 and thus become anemic, Okay, pernicious anemia. Pernicious anemia. Deficit of B12. Okay, so what might we see in our animals? We might see vomiting, diarrhea, weight loss, weakness, as you can imagine. We could diagnose this via fecal flotation. Okay, again, the eggs are not typically laid in baskets with this one, as with most of our parasites here at the end that we're covering. So the way we avoid it is just don't eat those intermediate hosts. Here you go. What do you guys notice about this egg? What's its shape? Oval. Very good. What about its color? Well, don't pay attention to color. You may say yellow, but it is typically a pale yellow. However, if your microscope light is an LED or something, it might make it look a little different. But I want you guys to look at that top end of that egg. What do you see there? It looks like a little hat maybe, a little topper. It's called an operculum. Okay, an operculum. So you've learned about bipolar plugs, right? Well, this is not a plug. It's not a knob up at the end. It's more or less what we would refer to kind of as a lid. Okay, not that it opens up and closes like a ketchup bottle, but it's going to be a little cap sort of a thing up at, on one end of the parasite, and it's called an operculum. An operculum, and it's only located at one end of the parasite. All right, we're going to see a lot of that in these upcoming weeks. What about the center material? Does it take up a lot of the shell or not a lot? A lot. Is our shell thick or thin? It's pretty thin. Is it smooth or rough? Hope you guys are getting the point here. Spiometra. I think this is our last one. Yeah, it's our last one. So hang in there with me. Spiometra. This is another pseudo tapeworm, okay? Its common name is the zipper tapeworm. So Spiometra species is known as the zipper tapeworm. The definitive host is a cat or a dog. It has also the two-stage intermediate host. So first there has to be an aquatic crustacean that then a fish or frog eats part of, the infected part of, and then they get infected, and then your dog or cat eats the fish or frog. Okay? And since this is a two-host intermediate, or requires two hosts, that parasite must undergo two transformations before it is infective to the pet. So if your dog eats the aquatic crustacean before the fish or frog gets involved, it should not get infected because that second one is required. So if you guys see, excuse me, I keep bending down because I have a little kitten attacking my ankles right now. If you guys see this egg on a sample, then make sure to look at the details about this parasite, because look, it's common to Florida and the Gulf Coast region. None of the parasites that you have learned about so far have had an area that they are specific to, okay? The diagnosis is going to be very similar to our last one we learned about. Do you guys look at that egg? What's the shape? Oval. Smooth or rough? Smooth. Center material takes up most of it. And this one looks like it's allowing a lot of light through, but in certain views it may not allow a lot of light through. But look at the operculum. You see it there at one end? One end has that operculum right over here. Okay? This end has more of a knob on it. Okay? This is not an operculum over here. This one is. All right? All right, so let's review. What are you going to tell the owner if they call in and they say, oh my goodness, I found this on my dog? You tell them, it's okay, we can treat for it. First off, have you seen any fleas at your house? We need to get those under control. And then come into the clinic, we need to get a current weight on your dog, and we can get them some medication. So how do they prevent reinfection? Yes, they get rid of the fleas if we're talking about Dipylidium caninum. What if we're talking about Taenia taeniformis? Very good. Limit hunting. You need to limit hunting, okay? So can their kids get this parasite? Whether it be any of our cestodes we've learned. Yes, typically they can, but again, they have to get it by an intermediate host. Okay, so this one's more similar to, say, our heartworms, where they'd have to get bit by a mosquito, rather than our hookworms, where they could just step on poop when it's the ovas have hatched into larval form. So can the other pets get this parasite from the infected pet's poop? No, they cannot. Can they get it from grooming the pet that has it? Possibly. If the pet that has it still has fleas, then yes, it is possible that they could get it from the other pet. So what symptoms might they see in their pet? Let's say if it's Diploidium caninum. I hope you're kind of sitting there like, They may not see any symptoms. They may see no symptoms whatsoever. What they are going to see is those little rice segments in the poop. Okay? All right, guys, that concludes this week's lecture. Thank you for being patient with me. Thank you for understanding why we had to cancel class because the district closed campus at the end of this week. Make sure you check with Rachel if your kennel duty is still meeting, if you have a Thursday kennel duty. And I will see you guys virtually online next week, okay? Don't forget to take the quiz. Thanks so much. Have a great afternoon, evening, or morning whenever you're watching this. Bye.